Hello everyone, welcome to this tutorial. In this demo, we'll show you how you can isolate, count and freeze PBMCs. We'll be working with 10 ml of EDTA blood. Remember to always balance the centrifuge before any centrifuging. We intend to take out the plasma first, and therefore we will centrifuge the blood at 445 Gs for 5 minutes. After the 5 minutes have elapsed, bring out the centrifuge bucket, spray it with 70% ethanol, and open it inside the safety cabinet. Take out the vacuum tube and proceed to harvest the plasma. Put the plasma in 2 ml Apex tubes for storage. To separate PBMCs from blood, we will use lymphocyte separation media. In this demo, we'll use LymphoPrep, which we will aliquot 4 ml into two 15 ml centrifuge tubes. This represents 40% of the total volume of our blood, and therefore, each tube will have 2 ml of the LymphoPrep. We need to replace the plasma we harvested earlier, and to do this, we add cold RPMI 1640 to our blood. Mix the blood gently until it is homogenized. In the absence of RPMI 1640, one can use PBS or HBSS. Take 5 ml of the blood and carefully layer it gently on the LymphoPrep in the centrifuge tube. Ensure that you hold the centrifuge tube at a 45-degree angle. Hold the serological pipette at the tip of the tube. Take the remaining 5 ml of the blood and lay it on the second centrifuge tube containing the LymphoPrep. In the absence of LymphoPrep, one can use Phycoplec or Histopec as lymphocyte separating medium. Centrifuge the samples at 450 Gs for 20 minutes. After the 20 minutes have elapsed, bring the samples back to the biosafety cabinet to harvest the Buffycoat, which contains the PBMCs. The Buffycoat lies between the RPMI 1640 and the LymphoPrep. Now, there are two ways in which one can obtain the Buffycoat. You can remove the RPMI on top gently without disturbing the Buffycoat. Discard the RPMI and proceed to take out the Buffycoat and transfer it to an empty collection centrifuge tube. Alternatively, you can go directly to the Buffycoat and carefully pipette out the cells. Cell harvesting should be done as fast as possible, as lymphoprep is toxic to the cells. After harvesting the cells, there is need for washing them so as to remove any traces of the toxic lymphoprep that will harm the cells. To wash, top up the cells up to 10 ml with cold RPMI 1640. Proceed to the centrifuge and centrifuge the cells at 450 Gs for 5 minutes. After the 5 minutes, bring back the cells to the biosafety cabinet for the second wash. One can clearly see the cells at the bottom of the centrifuge tube as a pellet. Carefully pour out the supernatant and gently dislodge the pellet in the tube. Add 10 ml of cold RPMI 1640 to the cells and mix well. Take a drop of the mixture and put it in a 96 well culture plate for cell counting. Proceed to the centrifuge to wash the cells at 450 Gs for 5 minutes. Cell counting is important as it informs us on the amount of nourishment the cells need before storage. Take 10 microliters of trypan blue and load it to a well next to the well that contains the cells on the culture plate. Mix the cell suspension well. Add 10 microliters of the suspension to the well containing trypan blue. Mix gently and load it on the hemocytometer. Observe the cells on the microscope. The hemocytometer comprises of large squares that are further divided into grids. The center grid contains 5 by 5 boxes that are further divided into tiny grids. The center grid is surrounded by 4 large boxes. Each large box is further subdivided into 16 smaller boxes. We will count our cells in the 4 large boxes that surround the center grid. To get the total number of cells, we use the formula number of cells counted multiplied by the dilution factor multiplied by the volume multiplied by 10 raised to power 4. The dilution factor is 2 because we diluted our cells with 10 microliters of trypan blue. We'll use a volume of 10 ml as this is the volume in which we suspended our cells before the wash. After the 5 minutes of the second wash have elapsed, wash the cells again for the third time using cold RPMI. and spin it for 5 minutes. This will be our last wash. After the last wash, discard the supernatant, distrap the pellet gently, and add 500 microliters of fetal calf serum, FCS, for nourishment. In the absence of FCS, one can use FBS, which is fetal bovine serum. Incubate the cells in ice for 30 minutes. Freezing media comprises of 20% dimethyl sulfoxide, DMSO, and 80% of fetal calf serum. Mix well the contents and filter it for sterility. After the 30 minutes of incubation have lapsed, take the cells back to the biosafety cabinet and add 500 microliters of the freeze mix to the cells. Mix well and transfer the cells into cryovials and place them in a Mr. Frosty freezing chamber. The Mr. Frosty contains isopropanol. Take the Mr. Frosty into -20 freezer. Do this quickly to avoid death of cells, as the DMSO is toxic to the cells. After 1 hour, transfer the Mr. Frosty from -20 freezer to the -80 freezer. Let the Mr. Frosty stay in the freezer for 24 hours. After 24 hours have lapsed, transfer the vials containing our cells to liquid nitrogen for long-term storage. Handle liquid nitrogen with care. And that's the end of our demo. Thank you everyone for watching.